Brain scans of people with severe cocaine addiction show dramatically reduced glucose metabolism in the prefrontal cortex, the region responsible for judgment, planning, and impulse control. This is not damage caused by cocaine, though that is part of it. It is the brain's own response to chronic drug use. The prefrontal cortex, overwhelmed by the distorted reward signals, progressively withdraws from regulating behavior it can no longer override. Addiction is not a failure of willpower. It is a disease of the organ that produces willpower. One of the most important discoveries in addiction neuroscience is the transcription factor delta Phos-B, which accumulates in the nucleus accumbens with repeated drug use and acts as a molecular switch, altering the expression of dozens of genes involved in reward, motivation, and synaptic plasticity. Unlike other proteins that degrade quickly, delta Phos-B is exceptionally stable. It persists for weeks to months after drug use stops. It appears to sensitize the reward circuit to drug cues while simultaneously blunting responsiveness to natural rewards, creating the motivational landscape of addiction at the molecular level. Addiction researchers Terry Robinson and Kent Berridge proposed the incentive salience theory of addiction, distinguishing wanting from liking. With repeated drug use, the dopamine system becomes sensitized to drug-associated cues, the sight of a needle, the smell of alcohol, familiar locations, such that these cues trigger powerful wanting and craving even when the person no longer particularly likes the drug's effect. This dissociation between wanting and liking explains why addicts in recovery can experience intense craving for a drug they intellectually understand has ruined their life. The wanting is not voluntary. One of the most consistent neuroimaging findings in addiction is reduced activity and reduced gray matter volume in the prefrontal cortex, particularly the orbitofrontal cortex and anterior cingulate cortex, which are involved in decision-making, impulse control, and the evaluation of long-term consequences. This prefrontal hypofunction means the addicted brain has literally less capacity to inhibit drug-seeking behavior in the face of cravings. The balance of power shifts from deliberate, goal-directed behavior under prefrontal control to habitual, automatic behavior driven by subcortical reward circuits. The heritability of substance use disorders ranges from about 40 to 60% across different substances, a substantial genetic contribution. Specific gene variants affecting dopamine receptor density, opioid receptor sensitivity, and stress hormone regulation all influence addiction vulnerability. But genetic risk is not deterministic. It increases the probability of developing addiction given exposure to substances, but environment determines whether that exposure occurs and how it is managed. Growing up in a household with substance users, experiencing trauma, and lacking social support all dramatically amplify genetic vulnerability. The ACE study, Adverse Childhood Experiences, tracked the health outcomes of over 17,000 Kaiser Permanente members and found a powerful dose-response relationship between adverse childhood experiences, abuse, neglect, household dysfunction, and substance use disorders, mental health conditions, and physical illness in adulthood. People with 4 or more ACEs had a 7-fold increased risk of alcoholism and a 10-fold increased risk of injection drug use compared to those with no ACEs. Trauma in childhood alters the developing stress response, HPA axis reactivity, and prefrontal development in ways that increase addiction vulnerability. Medication-assisted treatment, or MAT, is the evidence-based approach to opioid use disorder, combining medications with behavioral therapy and support services. Buprenorphine is a partial opioid agonist that occupies opioid receptors without producing the intense euphoria of full agonists, dramatically reducing craving and withdrawal while allowing near-normal function. Methadone is a full opioid agonist with a very long half-life that stabilizes opioid receptors. Both reduce overdose deaths, criminal activity, and disease transmission by 60 to 70%. Naltrexone blocks opioid receptors entirely and is effective for motivated patients who have completed detoxification. Cognitive behavioral therapy adapted for substance use disorders focuses on identifying and managing the triggers that produce drug craving, developing coping skills that provide alternative responses to stress and negative emotion, and restructuring the cognitive distortions that maintain use, such as believing that one is powerless to resist or that life without the substance is not worth living. CBT for addiction has strong randomized trial support across multiple substances and is particularly effective at preventing relapse by teaching skills that remain available after treatment ends. A vital but underappreciated finding from epidemiological research is that the majority of people with substance use disorders eventually achieve sustained recovery, often without formal treatment. The National Epidemiologic Survey on Alcohol and Related Conditions found that approximately 75% of people with alcohol dependence are in sustained remission at any given point. Recovery is facilitated by natural developmental processes, maturing out, as well as major life changes like marriage, parenthood, and employment. This data does not diminish the need for treatment, but it does counter the nihilistic view that addiction is a permanent, irremediable condition. The neuroscience of addiction should produce compassion, not contempt. When we understand that addiction involves measurable changes in prefrontal function, reward system calibration, and gene expression, not simply weak moral character, the appropriate response is treatment, not punishment. The science also provides hope. Brains that changed under the influence of chronic drug use can change again in recovery.